This is Breakthroughs, a podcast from Northwestern University Feinberg School of Medicine. I'm Erin Spain, host of the show. There is still an urgent need for more approaches to treat SARS-CoV-2 infection, especially as the virus mutates and produces more variants. Since early last year, scientists around the world have been studying angiotensin-converting enzyme 2, known as ACE2, as a potential therapy for COVID-19 because the virus enters cells in the body by attaching to ACE2 receptors. Daniel Batlle, a professor of medicine in the Division of Nephrology and Hypertension at Northwestern, has been studying ACE2 and its potential therapeutic uses for many years. When the pandemic began, he proposed a hypothesis that soluble ACE2 could treat the SARS-CoV-2 virus and lead to full recovery and survival. And now he has some exciting preliminary results to share. Thank you so much for coming on the show.
Thank you, Erin.
As a nephrologist, tell me how COVID-19 involves the kidney directly. I know your work with kidneys is why you were studying ACE2 to begin with. So talk to me a little bit about COVID-19 and the kidneys and how in some cases COVID-19 has led to acute kidney injury.
Very good question, and one that is very interesting, that the early reports about this disease all came from China, just because they were affected before anyone else. So if we go back to the first reports in February, March, April of last year, they were describing a disease as a purely almost respiratory disease, which indeed, that's what it is. But in the middle of 2020, when the United States was hit so badly, and some parts of Italy and Spain, it became clear that acute kidney injury is a major component of the disease. And as you probably read in the press, in New York, they didn't have enough dialysis equipment to treat all the cases. They were inundated because of kidney disease. In very ill patients, the incidence was estimated to be as high as 50 to 70%. And not only that, the best marker of bad outcome was kidney disease, not lung disease, but kidney disease. Well, both pretty bad, of course, but in the studies that were published, having acute kidney injury is really the bad thing that can happen when you have COVID. And again, we don't have a treatment for the kidney disease other than supportive care and dialysis. So yeah, we need to find a better treatment for the acute kidney injury. And it's also interesting that it's more than acute kidney injury. It's also a lot of protein in the urine, blood in the urine. It's not just the typical bread and butter type of AKI. Many patients develop a disease that's called collapsing glomerulopathy that causes massive proteinuria, protein leakage. And they have been CDs, some from here, from Northwestern, reporting this complication that affects mainly African Americans, by the way. So we need a treatment for the kidney disease associated with COVID as well as COVID. But of course, if you could treat COVID, then all the other complications should not develop.
There's still a lot of mystery out there when it comes to COVID-19 and how it affects different organs in the body.
Yes, and you asked me about invading the kidney. Well, that's still a very controversial issue. It's very difficult to demonstrate the virus in the kidney, which has been really a perplexing situation because the kidney is very rich on the receptor, ACE2. Yet, there have been many studies. Actually, we have a review with Louise Hasler, who is the main author of the paper that just came out, trying to review the evidence pro and against kidney invasion directly. And I can tell you that there are studies that show it very convincingly, but the majority cannot find evidence that the virus itself is present in the kidney. So that's something that's still the subject of a lot of research and interest. It's so easy to demonstrate the virus in the lungs and the lavage of the bronchial secretions in the kidney is very difficult to demonstrate. So it's possible that the virus somewhat elicits an immune response which causes kidney damage, but it's not very clear that's the direct invasion of the virus that causes the kidney damage. We need a little bit more of research in this area still.
You're a nephrologist and a physician scientist who, as I mentioned, has been studying ACE2 for some time. Tell me, how did you first become interested in studying ACE2?
So as a nephrologist, like many others, we've been interested in the renin-angiotensin system, ACE inhibitors, angiotensin II blockers. Is kind of part of what we do every day as part of our specialty. And ACE2 was the new player. So we've been working on ACE2 just because it was the natural thing to do as the extension of our previous work.
What was known about SARS-CoV and ACE2 before this current pandemic began?
Actually, the key pieces of information were already available from the previous epidemic that was confined to China with the coronavirus that known as COVID-19 or COVID that caused this epidemic. So back then, it was already known that the receptor for this coronavirus was ACE2. And there were elegant studies, but then the epidemic was fortunately went away and kind of the information was there, but many people did not make the association until January of 2020. And of course, the first person that recognized that was Fang Li, a scientist in Minnesota that had been working with SARS-1. And since we were working with ACE2, back in January, we made the connection right away and start working full speed with this, with the idea, providing more ACE2 could be a way to fight the new coronavirus. The critical information was there, but needed to be translated and applied to the new coronavirus.
And as you said, research on this enzyme just exploded because of COVID-19 and ACE2, that connection. There really wasn't a ton in the literature. You had been publishing some things, but just in the past year, there's been much published. So what do we know now about ACE2 and COVID-19 that we didn't know when this virus first emerged?
Well, it has been a mattering pace because everyone in the world has been working on the area. And the first studies very early in 2020. Back then I was following every day, every new paper that was coming, many preprint. But first of all, the receptor binding domain of the spike of the virus, the matches the ACE2 was recognized. Then there were studies that described the molecular structure, the interaction with ACE2. It was a very rapid pace. And everyone working on it, there was also a tremendous interest and a bit of confusion because people were confusing the ACEs and ARBs that all the patients seem to be taken. Could they amplify ACE2? Could that be a bad thing? And there were editorials right and left. And for the past year, there have been literally 1,000 papers debating whether these ACEs and ARBs are bad or good in the context of COVID-2. And now, after a year, the idea is that they are neutral, they don't do any bad things. And actually, there are studies emerging that suggest that not only you should continue taking your ACE and ARB if you are taking it, but that actually can help in some cases.
And again, those ACE inhibitors, they're really common. As you said, many patients are on these to lower blood pressure, prevent heart failure. How common is it that folks are on these ACE inhibitors?
Well, it's a standard of care that if you have chronic kidney disease, you should be in one of them. If you have chronic heart disease, you should be taken one of them. And for people with hypertension, I would guess that 50% of people with hypertension take one of those ACE inhibitors. And because initially it was found that hypertension, diabetes, heart disease are risk factors for the severity of COVID disease, the question is, what do we do? Shall we continue these drugs or shall we stop them? And now the information a year later is available that they were not going to do anything bad, as finally it has been shown after a year of research and publications.
So while all of that publication and really focusing on those ACE inhibitors was happening, you were still looking at this hypothesis that you suggested at the beginning of this pandemic. And explain to me that. Explain the concept of using soluble ACE2 as a decoy for a potential therapeutic approach to COVID-19.
The idea is very simple, that the virus, the spike of the coronavirus, looks for the ACE2 receptor. The ACE2 is in the cell plasma membrane. where ACE2 is most abundant. In the circulation, the levels of ACE2 are very low. So normally what happens is that the virus is looking for the receptor, and the idea is to therapeutically intervene by giving a sufficient amount of the available ACE2 protein, and we can talk about what type, but the idea started with the natural native ACE2 protein. So if you give enough of it, you are going to intercept the virus from reaching the target, which is the ACE2 that's in the cell plasma membrane. It's like giving a bait when you go fishing, so that the virus gets distracted and occupies the exogenous ACE2 that you are giving, rather than going to the cell membrane. Because the moment that it gets in the cell membrane, it gets internalized with ACE2, and that's how the virus starts replicating inside the human cells.
So tell me about the work that you're doing in your lab and some recent publications. You just published a paper about the ACE2 soluble proteins showing that it could neutralize SARS-CoV-2 in an animal model. Please tell us about those results.
Before the animal model, we've done extensive work along this past year in different cell types, cells that are susceptible to be infected by this virus. And the first requirement is that it has to be a human cell line. And by extrapolation, when you work with experimental models, the mice, for instance, cannot be infected unless you introduce the human ACE2 protein, which is a bit different from the mouse protein. So you did an animal model, and this animal model is now available commercially, but it took some time to get it. And everybody was online trying to get the same animal. I think we were among the very first to get it. Here we had it back in July. And it's an animal that, when you give a sufficient viral load, develops a fulminant disease. And by 7 days, they are all dead. Or they have to be sacrificed for human reasons, because all these protocols have to take into account you don't want these animals to suffer unnecessarily when the outcome is already known. Universal mortality, day 7 to 8 to 9. So we had this model in our lab, but we could not do the experiments here at Northwestern because you need a safety facility. And for this, we had to go and do the studies at the level 3 safety facility that's available at the Ricketts facility of the University of Chicago. And we developed our own protocol, and using one of our proteins that I can tell you more about it in a minute, these animals were infected, and sure enough, they were reporting to us every day the status of the animals. And, it was one of these experiences of a lifetime when the technicians were telling us, your animals are looking great, and all the other ones we have to sacrifice because they are dying. So before we had all the data to analyze, we already knew that we had something unique. So that's the animal model, and we gave one of the several soluble ACE2 proteins that the lab has been working with over the last year.
How was the human soluble ACE2 variant created in your lab and administered to the mice?
All the credit goes to a scientist called Jan Wasocki, who's done all this work. We started with short ACE2 variants for kidney disease, which is the focus of our lab. And having this protein, and by the way, Northwestern has 2 or 3 patents on this protein. So having this short protein, then the question was to improve it and modify it for the use for COVID. And we have accomplished 2 criteria that are essential. 1, extend the duration of action. Why? Because the virus doesn't take holidays. And if you use the natural protein that has a half-life of less than 8 hours, you would have to give it 3 times a day to ensure the protection. So we modified our protein to confer this enhanced duration of action using a tag that we call albumin binding domain that increases the residency time of the protein from just a few hours to several days, at least 2 or 3 days. So we published that, and we were happy until more recently that we realized that this protein could be improved further in terms of binding power for the spike of the coronavirus. Jan Wasocki did another modification in the protein, created a link that causes the dimerization of the protein. Normally it's a monomer, and in this dimerized form happens to have maybe a 20-fold increase in binding power. So we think this feature is going to be important because the amount of protein that needs to be given depends on the binding power, and having an excellent binding power, we think we'll be able to treat patients eventually with a dose that is not as high as it would be otherwise. So we have a modified protein that simply is optimized, and we still are working in ways to optimize it further, although I have to say we are very happy with what we have right now.
Well, you mentioned it was sort of a once-in-a-lifetime experience when you were getting that feedback back from the lab that was watching the mice. Again, just explain to me how that felt to hear that this was working.
Well, the people that work in this facility go in a couple of times there, and they have a score to see how active is the animal, what's the daily weight, and all that, by the way, is in the publication. And they are able, by experience, to see whether the animal is very sick, terminally ill, and that's all they could report to us. And of course, you are just excited when you hear the report that your animals are surviving, and not all the research we do has that kind of gratification. Because if it works for mice, it will work for humans just as well.
And again, you've released these results in a preprint publication. Tell me about that.
Well, I hate preprints, I have to say. I am more of the classic, wait for the reviewers. But here, you don't want to wait 2 or 3 months for the paper to be published, because I think that the world needs to know, and I think this protein has to be created for human use, and the sooner the better.
Well, and you said you really wanted to get the word out about this, because while we do have vaccines, there are therapies that are being used for COVID-19. There's still a need out there as these new variants continue to appear. Talk to me about that and why it's important to look for other therapies right now?
Well, first of all, there has never been an effective therapy for COVID except for the vaccines. And we rely on the vaccines, and it's the solution to the pandemic. But there has never been a totally effective treatment. There are things that provide some marginal benefit, call it steroids, call it antivirals, but nothing has been completely effective. So definitely there is a need for effective treatments. The larger question is, if we control the pandemic, we will not need to treat anyone. But we all know that, number one, not everybody takes the vaccine. Some people are resistant, maybe 5% of them. There is the feeling in some circles that the disease will be controlled, but it will become not the disease of the pandemic, but just another disease like flu. For the cases, if we can find an easy way to give the protein, and if it's proven to be effective, then you'll have another treatment that right now is not available. That is not to say that someone may come up with an antiviral one of these days that's going to be as effective as the antivirals were for HIV. Hopefully someone will. But in the meantime, what we have theoretically, in my opinion, is the very best you can aim for. And again, it's all limited to the results and experience with these animals that we were able to infect and cure recently.
When could it move from the animal model into clinical trials?
Well, there is a clinical trial, but with a different protein, the native soluble ACE2 protein, and the results should come out soon. I hope they are positive, but I wouldn't be surprised if they are not as great as they could be. Time will tell. Number one, because the soluble ACE2 protein is given systemically in the veins, and I think it's much better to give this protein, as we did, intranasally directly to the lungs. And not to mention that I think we have an improved protein based on the characteristics that I described earlier in terms of duration of action and improved binding capacity. So what do we need to do with our protein? We need to manufacture it and get an IND so that it gets approved through an expedited pathway, and that's where we need a lot of help.
Back in 2003 with the first SARS-CoV, this is when ACE2 was discovered to be the host receptor. But there could be other coronaviruses in the future that pop up where this technology, this protein could all be used again.
Absolutely. Not all the coronavirus, but some of them use exactly the same receptor, ACE2, and have been prepared for the new outbreak. with new coronavirus, that alone justifies the research and the existence of this protein in the market.
Well, thank you so much for coming on today and sharing this latest research, which sounds very exciting. We're looking forward to seeing what happens next.
Thank you.
Thanks so much for listening to the show. You can always head to our website, feinberg.northwestern.edu, to listen to past episodes of the show, claim CME credit, and subscribe. Subscribe on Apple Podcasts or wherever you download your shows so you never miss an episode. Thanks for listening.